Alrighty, welcome everyone. So today we're talking about comparative genomics. So here we are towards the end of the term. So we talked about electronic health records last time, and we have two lectures on comparing genomes, and then we have Thanksgiving. So today's lecture is focusing on the conserved parts, and then Tuesday's lecture is going to be focused on the changes, how things change. And then we have two lectures on metabolic modeling, and then your final presentations. So let's dive right in. So we've talked a lot about comparative genomics in the alignment sections. So our goal was to measure conservation of functional elements. And the key idea was that by studying the alignments that we were making in lectures 2 and 3, we are now able to pinpoint evolutionarily conserved nucleotides. Everybody with me so far? So those evolutionarily conserved nucleotides, namely the places where the alignment actually finds matching positions, are also the ones where functional elements lie. And the reason for that is because of mutations being subject to selection. If a mutation disrupts a functional element and decreases fitness, that mutation will eventually make it out of the gene pool. The remaining ones will be preserving the remaining ones will be preserving the function, and therefore they will be preserved over evolutionary time. Okay? So we can read evolution to reveal functional elements. So today we're going to be talking about the part where we use evolutionary conservation to understand genomes. And then on Tuesday, we'll talk about how we can use genomes to understand evolution. Everybody with me here? So we're going to talk about different stages of comparative genomics. The first is studying nucleotide level conservation and measuring evolutionary constraint based on whether the nucleotides have been preserved. The second one will be looking at evolutionary signatures. And there we're going to be focusing on the patterns of change rather than the overall conservation. And we're going to look at such signatures for protein coding genes, and then for RNAs, motifs, regulatory sites, and microRNAs. And then we're going to talk about how do we use all of the modern deep learning methodologies to go beyond just simply nucleotide level conservation and beyond pattern conservation to functional conservation. So we can basically measure conservation at the primary sequence level, or you can measure conservation at the level of selection by observing patterns of change, or you can measure conservation at the level of predicted function using all of the regulatory genomics, convolutional neural networks, deep learning functions that we've talked about. Or you can measure constraint at the level of not just the activity of a single element, but the predicted activity of the group of elements together acting on gene expression. Is everybody with me here? So the key idea is that we can get at something that's closer and closer and closer to what selection actually is acting on. And if you change a single nucleotide in a protein sequence that doesn't change the protein translation, selection is blind to that change. And therefore that change will be tolerated. Everybody with me here? And similarly, if you change something that changes the nucleotide, but actually preserve the function of the motif, for example, you might be tolerant to that. Or if you change the whole motif, but you preserve the function of the enhancer, you might be tolerant of that. And if you change the enhancer, but you preserve the function of gene expression levels, then you're tolerant to that. Is everybody with me here? Can I get a 5, 4, 3, 2, 1 as to who's with me? Awesome. Awesome. So basically the key idea is that we can measure either primary sequence conservation or conservation at these higher levels of abstraction. And we've been talking a lot about this concept of measuring selection at the level of inferring function, of basically this representation learning paradigm and transformation of deep learning and modern generative AI and so on and so forth, has been about these increasingly higher levels of abstraction. And it's the same thing here. We can basically do evolution here or at the signatures, at the patterns, at the regulation, at the expression. And you can do all of that for primary DNA sequence, but you can also do all of that for protein sequence. You can measure evolutionary conservation at the protein level, or you can say how many of the amino acids are preserved, or you could score different amino acids based on the observed frequency of substitution. Is everybody with me here? So you can basically, in the BLOSSOM matrix, for example, you're looking at how frequently is a particular amino acid replaced by another amino acid. So now you're going to score that in a different way. And you can do the same thing with iterative searches, where you can basically say, okay, this is for all proteins, but for this one protein, I can basically run my search iteratively, find what's conserved, refine it, and then go and search for that one protein. Or you can go a step further, and we talked about protein language models, where you can basically model the function, quote unquote, of an amino acid in the context of all domains that look like this one, because you've now modeled the language of protein structure and the language of protein sequence. So you can now start measuring conservation at the level of protein language models, but you can also measure conservation at the level of protein function, which goes beyond just protein language models. And then again, the question is, can you find the right level of abstraction, the right level where evolution is actually active? Okay? Who feels that they're learning stuff? 5, 4, 3, 2, 1? Awesome. Great. Awesome. So let's dive right in. So basically, first, let's look at nucleotide level conservation and how to measure evolutionary constraints. So you can basically do comparative genomics. You can align species across mammals, across flies, across fungi, and so on and so forth. And then the key idea here is the more branch length we have, the more power we have. Namely, if I align only a pair of species, all I have the option to do is go further in evolutionary distance. So if I'm trying to align something with human, for example, you can basically say, oh, well, I can align something with chimp, or, I can go further with, mouse and rat, or I can go all the way to, I don't know, armadillo, okay? Tell me the difference between the power of comparative genomics when aligning human with chimp. How much power am I going to have to discover conserved elements? So we'll have very little power because the vast majority of the genome is conserved. And therefore, I don't have enough events to measure divergence, okay? If I now go all the way to, I don't know, armadillo, then I have more power, I have more events. However, armadillo is actually quite different from human. And therefore, yes, I have more signal. Yes, I have more ability to distinguish the non-conserved parts, but I've actually started losing my ability to distinguish conserved parts, okay? So that's what takes us to branch length. And you can basically say, I can go to very, very distant species, but by the time I get there, not much is conserved. So yes, I've eliminated the noise, but I've also eliminated my signal. Or I could basically say, well, why don't I increase the number of species? And that allows me to stay at close evolutionary distances, but then accumulate more spurious, more mutations in non-functional regions. So basically my noise will be better eliminated if I accumulate species, okay? So more branch length give us more events and also more power. And if our goal is to distinguish functional versus non-functional based on the number of mutations, very close distances have no mutations in either of the two, so that's not good. But with sufficient distance, you increase the ability to distinguish them. But at very far distances, the functional regions are no longer conserved. By contrast, if you use many closely related species, that's much more power than few distantly related species, because for the total branch length, you prefer many close species because the functional regions are conserved in every pair of species, and the non-functional regions accumulate the noise independently. The analogy that I like to give is that you're recording an orchestra with multiple microphones. Surrounding each microphone, you have some amount of noise, but the noise is independent in each microphone, and therefore you're more likely to capture the true signal. So So you can think about that signal being, the functional elements, and then you're measuring it from many different angles, many niches, many species, each of which accumulates the noise independently. Everybody with me here? Okay. So again, we can basically distinguish different types of events. We can think about nucleotides only, and we can basically say how many edits have there been, how many substitutions, where every amino acid or every nucleotide substitution counts the same, and you're just measuring edit distance. Everybody with me here? So that's the simplest model. But we already saw much more sophisticated models. We could basically say, well, A and G are replaced much more frequently, and T and C are replaced much more frequently, are exchanged much more frequently than either of them with T or C. And the reason is just simply because of the mutational distance where DNA polymerase confuses them more frequently. And therefore, I can use that from the mutation process alone to basically talk about the fact that if I see a T on top of a C in an alignment, it's more likely to be from the same ancestral sequence. Okay? And that's just at the mutational process. Now, at the selection process, there are regulatory motifs, for example, that are more frequently switching T with C. And also within protein coding regions, the third codon position is more frequently exchanging T with C or A with G while preserving the amino acid translation. So that basically means that I can, across different species, measure the frequency with which nucleotides are replaced and then get a different score for different types of replacements. And this is much more powerful in protein sequences, where the amino acids that are frequently replaced are amino acids with similar properties. And therefore, you get a very negative score if you align two amino acids across boxes. But within each box, you basically have amino acids with similar properties. And therefore, the score of exchanging them is actually positive. That score tells you that you're more likely to be aligning true signal. Everybody with me here? Yeah? So basically, the way that these matrices are computed is based on a log likelihood ratio. That log likelihood ratio is between two different hypotheses, a related hypothesis and an unrelated hypothesis. Under the related hypothesis, you're simply asking for true alignments of true orthologous regions. What is the frequency with which these two amino acids co-occur? And then the denominator of unrelated sequences basically says, based on the individual frequencies of each amino acid, how likely am I to find them on top of each other? Okay? So that allows me to now have a probabilistic interpretation of that alignment based on that substitution. Okay? And all of this sits over here. So basically, I can look at primary sequence, or I can look at substitution frequencies that allow me to be a little more sophisticated about what the preservation is. Okay. Now let's talk about how we can, use that systematically, how we can apply it. So basically, the goal is we're trying to infer whether something is functional or non-functional based on the observations of different types of events, of different substitutions. And this is very similar to what we talked about in hidden Markov models and in, the very first lecture, where we said that there's a set of observations. These are the substitutions. And then there's an inference. This is a model, a hypothesis of whether something is coding or non-coding, okay? And the key idea here is that we can say, if it's conserved, what's the frequency of substitution? And if it's not conserved, what's the frequency of substitution? And what we're observing is that frequency. And then we're inferring the probability of conserved, not conserved, okay? So if we're trying to detect these conserved elements, the first thing is we use lecture 2 and 3, and we basically carry out alignments. So basically, I simply line up the nucleotides or the genes from one genome on top of the other genome. Once I have these alignments, I can basically use these to do comparative identification. Where are the functional elements? And we saw this picture before on the alignment lecture, where we basically said, I can go through and measure the amount of conservation, the amount of constraint at every region. And I can use different metrics for that. I can count the number of edit operations. I can estimate the number of mutations, And we're going to talk about that in the phylogenomics lecture, or in phylogenetics lecture on Tuesday. I can incorporate information about the neighborhood. I can think about windows. Or what I'm showing here, I can estimate the probability of being in a constrained hidden state of my hidden Markov model. So I can build a hidden Markov model exactly as we saw in the HMM lecture. And we can basically use that to say, how likely am I to be in a conserved state or in a non-conserved state based on the observation of the number of substitutions that I'm observing, okay? I can also use the phylogeny, as we're going to see on Tuesday, to estimate the mutations not individually in a column, but based on the tree, based on every single divergence event. And we can also allow different portions of the tree to have different rates, which we're going to look at in phylogenomics on Thursday, okay? So we can use that to distinguish neutral sequence from conserved sequence, to distinguish quote unquote non-functional sequence from quote unquote functional sequence. Okay? And that basically means that at every position, I can say, well, how many substitutions have there been? And if a sequence evolves neutrally, if there's nothing acting there, then you will expect some number of mutations. But if there's constraint to preserve a functional element, then you expect a decreased rate of mutations. Okay, everybody with me here? And then I can just simply measure the rate, basically the length of the branches of the overall tree, and therefore say, well, that's a long tree, probably neutral. That's a very short tree, probably constrained. But I want you to also realize that super long is not neutral either. In other words, if my tree is beyond what I would have expected for a neutral sequence, that might actually indicate positive selection. Is everybody with me here? So the next part is I want to go not just at the rate of substitution, but perhaps at the pattern of substitution. So let's look at this position here. I have a C in, say, the reference genome. In this particular case, the reference is mouse. But then in multiple other species, so every dot here means that the reference is preserved, okay? So if the reference is mouse here, that means that every dot has exactly the mouse sequence. And notice here that I have 5 T's in a row. Are these independent mutations? No, there is single mutation that happened in the ancestor of that tree. Sounds good? So I can also see that in this particular position, I have a G or a C, a G or a C. So I have, C here, G, C, 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 G, C, C, G, C, etc. So there's a bunch of species that have changed independently into a G. This is not phylogenetically consistent. So what's going on here? What's going on is that the sequence under constraint is probably only carrying that there's a C or a G at that position. And therefore, there's no additional constraint. If there's a mutation that happens there, it doesn't change the function. Everybody with me here? So that's an example of a pattern of change, not just the rate of change, but the pattern of change. So you can actually estimate a position weight matrix for every place in the genome that basically tells you what nucleotides appear to be under constraint at that position. And that's treating every one of those nucleotides independently. Everybody with me here? Awesome. So you could do that and apply it across the entire genome, and you can actually start reading off sequences directly from the genome. You could basically say, oh, well, look, at this position here, I'm preserving clearly a C, clearly T. But at this position here, I might be preserving, a degenerate position that says either C or G. And I can start building these position weight matrices that say here a T or a C are equally likely, and therefore there's less constraint for either one of them. Sounds good? And you can use that to actually start detecting individual regions of the genome that are under constraint. And you can basically say, okay, well, this region here appears to be matching a motif. And guess what? There's a mutation associated with a phenotype that changes this G into an A in the human genome, even though that G appears to be evolutionarily conserved. So that suggests that that mutation might actually be causal through disrupting that regulatory motif. Everybody with me here? Awesome. So we can use this to detect individual elements that are constrained, or we can use this to estimate the total amount of constraint. And that's very important for many, many different applications. You could basically say, how long is the tail of conservation, and how much can I observe as above expectation? And here you could say, well, what is the distribution of constraint, or at least of conservation, that I get in neutrally evolving regions? And what do I get in the rest of the genome? And what I can do is observe an excess constraint that basically says at every position, I find slightly more than I would have expected. Okay? And I can put different cutoffs. And at this cutoff here, at the very stringent cutoff, I capture a lot of the gray and a little bit of the white. So I capture a lot of the signal, but also some of the noise. Because at that level of constraint, I still have, let's say, 5% of the elements that are spuriously conserved. Is everybody with me here? If I'm less stringent, I'm including more of my signal, more of the gray, but I'm also including more noise. But I can estimate that. I can basically say, I know that here I'm including 5% noise, and I know that here I'm including maybe 30% noise, and I know that here I'm including maybe 50% noise. Okay? And therefore, I can have a different level of confidence associated with every threshold. At this threshold, I'm 95% confident that whatever I detect conserved above that is actually constrained, like, truly constrained. And here I can be maybe 60% confident. Everybody with me here? But I can also, even though I might not be able to detect which is which, I can simply integrate the difference between the two curves and say that I estimate that overall, there's, 7% of the genome that's under constraint. Even though I might not be able to detect all of it, because down here I'll have a 90% false positive rate, I can still say that given the positive rate and given the fraction of the genome that I'm expecting to be constrained, the false positive rate, then I can still estimate the total amount of constraint. Is everybody with me here? Can I get a 5, 4, 3, 2, 1 to how well you're following? Nice, a bunch of fives, this is great. Okay, so that basically means that I can now start asking, given, say, the pattern of constraint or the rate of evolution, I can ask, is there some excess constraint on one side of the curve? And what you see here is that if ancestral repeat elements that we can hypothesize might not be under constraint, these are just ancient repeats that are still sticking around and mutating roughly neutrally, I can compare that with the distribution that I get for the entire genome. And what do I see? I see, sorry about that. I see that the blue curve has a longer tail towards slower mutation. Everybody with me here? So the vector is, the rate of constraint is actually slower for the whole genome than what you see for ancestral repeats. In other words, there are more slow evolving elements across the entire genome than what I would have expected based on the distribution of rate for the ancestral repeats. That allows me to estimate the amount of purifying selection. This is selection against change. Okay? But on the other side, I also see that the red curve drops off before the blue curve. And this is perhaps evidence of positive selection. This is selection for change. Again, functional elements are both on the left and on the right. If something evolves way too fast, that's not just random. It's probably selection for change. Does that make sense? Awesome. So we can basically say, where is the constraint? What is the highest enrichment for constraint? And so on and so forth. So that's all measuring the rate of change. You can also measure the pattern of change, but you can also ask simply, is there an alignment? How many species are aligned at that position? And simply measuring the number of aligned species gives you an additional signal that for example, protein coding exons have on average, 25 of the 30 species aligned there. Whereas, ancestral repeats on average have only like 4 or 5 species, or, maybe 10 species aligned there. Do you see that? So basically just simply counting the number of species in my alignment already provides a signal that allows me to distinguish likely functional regions that simply have more species aligned there. Is everybody with me here? Awesome. And the question is, of course, how does your power increase with the number of species? So we went from 4 species to 29 species, now to 200 species. And then the detectable fraction increases, but also the estimated constraint sometimes will increase, but not as much, because you can still estimate that constraint using this approach, even when you don't have the power to detect every single one of those. Okay? So this is about nucleotide level conservation. Now let's switch to evolutionary signatures. This is basically asking not just how many of the nucleotides are conserved, but also what is the pattern with which they change. Okay? So the question is, yes, comparative genomics can help us reveal functional elements. Basically, you can say, okay, this region here is clearly conserved, but it's not a protein coding exon, or at least it's not annotated to be a protein coding exon. One possibility is that there is still an exon here. There's a protein coding exon here, a protein coding region here, that is simply not annotated in the reference transcript because it is alternatively spliced. But you can have additional patterns of conservation. For example, here you can say, well, this is only conserved within mammals, but not in chicken, in fish. And therefore, this might suggest that this region is not like the other protein coding exons that are deeply conserved. It could be a recently evolved exon. For example, exon 8 and exon 9, they don't show as much conservation in other species that are farther away. So, that might be one of those. But you could also say that maybe it's a non-coding regulatory element. Maybe it's a microRNA cluster. So the question is, can we use comparative genomics to pinpoint the specific function of a region? Can we study the patterns of change to distinguish different types of functional elements? And the reason, again, as I mentioned earlier, is that the specific function dictates where the selective pressures are acting, and therefore dictates the patterns of mutation, insertion, deletion, etc. Okay? Based on where the function is, the pressure is acting at the function. It doesn't care about the primary sequence. It's not blindly copying every letter. It is preserving the exact sequence of those letters that matter. And if the exact sequence doesn't matter, it will preserve what everything translates to the same function. So the question is, can we develop evolutionary signatures that are characteristic to each type of function? And you can see these signatures here. So if something is protein coding, then those mutations that preserve the amino acid will be tolerated, even if they disrupt nucleotide sequence. So we can distinguish protein coding genes based on the frequency with which different codons are substituted and based on the conservation of the reading frame. You can distinguish RNA structures based on the preservation of the pairing of the residues. And therefore, if these two residues are paired with each other and one changes, the other one better change as well to preserve the pairing. Everybody with me here? And then for a microRNA, as long as the pairing of those regions is conserved, the sequence at the tip of at the pin here or at the ends doesn't matter. So you basically have a very particular conservation shape. And similarly for regulatory motifs, as long as the position specific weight matrix, the PWM or the PSSM, is preserved, then the function, effectively, the binding of the regulator is preserved. Okay? So what we're going to do is we're going to develop these signatures for each class of elements, and then we're going to use these signatures to discover new protein coding genes, to revise the protein coding annotations, to detect unusual gene structures, to detect novel RNA structural families, to detect targeting, editing, stability, ribose switches, to discover new microRNAs, to find new regulatory motifs or individual regulatory motif instances, and so on and so forth. Okay? So let's dive right in. What are these evolutionary signatures? So here I'm showing you a transition between a protein coding sequence and a non-coding sequence. And your goal is to figure out which half of this sequence is protein coding and which half is non-coding. Okay? So what do we know about protein coding regions? How are they translated? Every how many nucleotides do I get an amino acid? Every 3. And what else do we know about the genetic code? We know that there's groups of amino acids, groups of codons that code for the same amino acid. So yeah, there's redundancy. There's redundancy. That basically means that because there's only 20 amino acids and there's 64 possible codons, there's groups of codons that code for the same amino acid. How are they arranged? So you have, for example, GAA, GAC, GAT, GAC, that all code for the same amino acid, right? What does that tell me about the substitutions? Will there be more substitutions in the first, the second, or the third position? The third position. Okay, now with that information, take a look at these sequences again. Do you see anything funny about them? So the first thing that you'll notice is if you look at the sequence on the right, the first 2 codon positions appear to be conserved, and the third one appears to not be conserved, okay? So if I color it this way, what you see is that there's this pattern where you have conserved, conserved, not conserved, 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 not conserved. So if you were playing a battleship, it's like bang, bang, poof, bang, bang, poof, bang, bang, poof. Is everybody with me here? So you see this periodicity of the mutations, right? So that basically tells you that maybe this is because this one codes for amino acids and this one doesn't. Notice also the length of the gaps. So here, the length of the gaps is 5 and 1. What happens if you have a length of 5 in an amino acid sequence? That means that all of the letters in the middle are going to be translated nonsensically. Whereas here, all of the gaps are multiples of 3. So here, a gap of 3 is tolerated because it only knocks out 1 amino acid. A gap of 2 is tolerated because it only knocks out 2 amino acids, but the reading frame of translation is preserved. Is everybody with me here? And then you can go a step further and say, what is the frequency with which specific triplets of nucleotides are exchanged? And then you'll see that the exchanges that happen here from GAG to GAA, for example, preserve the amino acid, whereas the ones that are occurring here are just not preserving. Is everybody with me here? So, and then you see that at the conservation boundary, you basically have a splice site, an AG acceptor splice site that is, also perfectly conserved. So that basically means that there's a transition here from non-coding to coding. And it's kind of interesting that you guys were leaning towards the left side because it shows more conservation. What do you think that is? That's because there's probably gene regulatory elements here associated with perhaps that splicing event. And there might be one here as well, where, but what's interesting is that on average, you would expect, to some approximation, protein coding regions to be conserved only at 66%, 2/3, because the third codon position might not matter that much, right? So it's kind of cool. By chance, you would expect more conservation in functional regulatory regions than in functional protein coding regions, right? Because that selection doesn't happen at the nucleotide level. It happens at the amino acid level, okay? And this is what we call evolutionary signatures of protein coding selection. Does that make sense? So who feels that they've learned something? Can I get a 5, 4, 3, 2, 1? Awesome. So basically, again, by looking more precisely at the patterns of change rather than the total amount of conservation, we get signal. In fact, I'm going to go a step further and say that here, we're not even paying attention to the conserved positions. We don't care. If something is conserved, it doesn't teach us anything. All we look at is the patterns of change. And this is what evolutionary signatures are about. They're not conservation signatures. They're evolutionary signatures where the change is what is actually informative of the underlying function. Because the more changes you have, the more events you have, and the better you can understand the shape of the selective landscape in that region. Okay? Cool? All right. And then the reason, of course, is that if you disrupt, for example, the fat, cat, sat, if you cut out one letter, F, you basically, the at makes no sense. You cut out FA, the ka, sat, makes no sense. But then if you cut 3 letters, the cat, sat is readable again. Okay? So that's why you need to preserve the mod 3 length of the mutations needs to be 0. Okay? Similarly, if you look at the genetic code and how it's arranged, so you have CAT, CAC. As I mentioned earlier, a T to C substitution is much more likely. An A to T substitution is much more likely, but an A to T or an A to C or a G to C or a G to T is less likely. And the reason is that they are more likely to disrupt the amino acid. But that also means that the third codon position of anything that starts with GC is completely unconstrained. Anything that starts with AC, completely unconstrained. Okay? So that basically means that for the vast majority of codons, the third position is simply degenerate. Doesn't make a difference. Okay? And therefore, if you only care about amino acid conservation, you don't expect more than 2/3 of the sites to be conserved. And now if you apply this in a region, you can basically color amino acids. So you can color codons according to their substitution patterns. And if you look at a protein coding region here, and you look at its alignment, and you color all of the places that have changed, you see that only synonymous changes have happened here. And if you color this region here, which is highly conserved, but not known to be coding for protein, then you see that there's many non-conservative changes that change the properties of the corresponding amino acid translation. Okay? That takes us back to this blossom matrix that I showed you earlier, where the specific frequency with which different amino acids are exchanged is dictated both by the mutational distance between them, but of course, much more so, especially at long evolutionary distances, by the selective and the functional difference between them. And therefore, when you're looking at these patterns of change, you basically see that these are the corresponding translation corresponds to a non-conservative amino acid substitution. Everybody with me here? And you can also look at the reading frame. And what you see is if you color nucleotides in this condensed alignment, where I'm not showing the gaps, but if there is a gap, I'm coloring them. If they're out of frame, basically see that the further out you go evolutionarily, the more reading frame shifts you also have. And you also have random stop codons appearing in the middle of these sequences. Okay? That basically means that I can color every position of a very long alignment, and I can just directly read off where are the protein coding regions. Can anybody see a protein coding region here? There's a giant one right in the middle. And then that entire sequence basically starts with a methionine and an ATG, and it stops with one of the stop codons. And you can see this stark difference between the non-coding and the coding patterns of change. Okay? So pop quiz, what color are conserved positions here? If something's conserved, what color is it? They're white. So I'm not even coloring the conserved positions. So you might say, oh, well, maybe there's more conservation here, or maybe there's more conservation there. But in this particular case, we're not even looking at the conserved positions. We're only coloring the changes, and the changes themselves are extraordinarily informative. So going back to what I mentioned earlier, whether you have an alignment or not is informative. Whether in that alignment there's nucleotide is informative, and what that nucleotide is informative. Sounds good? Awesome. So the second signature is one of reading frame conservation. So what does the reading frame conservation mean? That basically means that I delete entire amino acids at a time rather than one letter in a codon. And the way that you can measure that is by looking at what fraction of the nucleotide positions are still in the same reading frame. And you can, basically see that if something is protein coding, it's 100%. And if something is non-coding, it's much less than that. Okay? And you can measure that by choosing the reading frame that would show the most conservation. And on average, you expect to see, much, much less for neutral conservation. So you can use this to now go back and revisit the annotation of a genome. For example, in the yeast genome, there were 4,000 genes that were annotated with a name, suggesting that they have a known function. And you could take 300 intergenic regions that are unlikely to be protein coding. And what you can see is that this reading frame conservation test alone is able to have 99% sensitivity and specificity. But if you now take 2,000 hypothetical open reading frames, these are genes that were not previously named, what you see is that 500 of them are simply rejected by the test, suggesting that perhaps they're not actually protein coding. And what you see here is one such example of a rejected ORF where you don't see this periodicity of mutations that you see here. So over here in a protein coding region, you see conserve, conserve, not conserve, bang, bang, poof, bang, bang, poof, bang, bang, poof. Throughout, you see this periodicity of conservation. Whereas here, you see no such periodicity. You see a bunch of insertions, and you see, random stop codons. Everybody with me here? So you can basically build an evolutionary framework for studying this. You could say, can we build a matrix, a substitution matrix that tells us, here's how I expect protein coding regions to change, and here's how I expect non-coding regions to change. So we're going to build a rate matrix that basically tells us with what frequency our different codons exchange with each other. And we're going to build one such matrix for protein coding and one such matrix for non-coding. And based on the frequency with which 64 by 64 pairs of protein codons, of protein coding codons are swapped, you can say, well, the swapping that happens in this region is much more likely to be coding. The swapping that happens in that region is much more likely to be non-coding. Is everybody with me here? So just based on the frequency of exchanges, you can say this appears to be evolving like a protein coding region and evolving like a non-coding region. Okay, can I get a 5, 4, 3, 2, 1? So who's following here? Yes, I see some 4s. This is great. Okay, so we can now estimate what is the probability that this region, this observation at the leaves of this phylogenetic tree are in fact evolving under a matrix that is protein coding or non-coding. And for that, I have a likelihood ratio between these two that basically tells me that the probability with which this region is protein coding is 10 followed by 35 zeros more likely under a coding model than under a non-coding model. And this one is 10 followed by 20 zeros more likely to be non-coding than protein coding, because you can basically calculate the probability of observing that exact sequence of leaves under a coding model. And that's, of course, tiny because there's many, many such alignments. So it's 10 to the minus 275. And the probability of observing that exact sequence with a non-coding model is also tiny, 10 to the minus 254, but 20 orders of magnitude more likely under non-coding and 35 orders of magnitude more likely under coding here. Sounds good? So this is extraordinarily powerful. And that allows us to now do all kinds of things. We can basically go through a region and just evaluate what is not the nucleotide conservation, which we're showing here, but instead the protein coding conservation, which I'm showing here. And that allows you to now see that every single codon has extraordinary signal. And in addition, there's this exon here that appears to not be annotated, but is very clearly protein coding. Everybody with me? Who thinks this is kind of cool? Can I get like a thumbs up? This is amazing, right? You can just read off protein coding constraint across your sequence. You don't have to worry about, oh, is it more conserved? Is it less conserved? You can just, read it straight off. And then indeed, if you put a probe and you try to capture an RNA molecule with that sequence, you sure enough capture that whole transcript. It basically shows you that this was just an alternatively spliced form. And you can also discover completely new protein coding genes. The length of this one is, remember the lecture last time about death by round numbers and sharp thresholds? There's a sharp threshold at exactly 100 codons, with which people said, okay, we're going to annotate protein coding genes in the human genome based on 100 codons or more. Well, this was just below that sharp threshold at 95, and it shows it's extraordinary constraint, but it wasn't captured before. Okay. You can also now ask, for all of these hundreds of new protein coding regions that we have discovered, do they help us annotate disease regions? And indeed here, you see that this genetic variant associated with myopia, interestingly, we had somehow missed. We were blind to it for some reason. And that region was previously thought to be non-coding, but now the genetic signal you can see localizes exactly at this coding exon, which is expressed, very specifically in the retina of an adult and the eye of a fetus. Okay, so this is kind of cool. We can now start annotating non-coding regions to actually be coding, and we have 118 such locations associated with a diversity of traits that are now protein coding. You can do the same thing in other species. So we've done that for human, we've done that for Drosophila, we've done that for yeast. And we also did this for the COVID-19 SARS-CoV-2 genome. And what we found is that from the 29 hypothetical ORFs, 28 of them are extraordinarily well conserved, but one of them, ORF10, basically shows absolutely no protein-like constraint, suggesting, in fact, it is a non-coding RNA. Conversely, we found that this signature of protein coding constraint is there for two overlapping reading frames in the ORF3C and ORF3A region. And therefore, on top of one of the genes, we found another gene that is encoded in a different reading frame that is also a functional protein coding gene, and so on and so forth. You can also do something very weird. You can basically say, well, are there cases where there's a stop sign and translation doesn't stop there? So you basically have protein coding constraint continuing to run right past this stop sign all the way to the second one. So what's going on here? So there's this joke about this guy in Greece who's basically, running right past a stop sign. And the policeman says, excuse me, sir, you didn't see the stop sign? And the guy says, I saw the stop sign, officer. It's you I did not see. So in this particular case, it just runs straight past the stop sign. And what we found is that this is not just one case. There's 300 genes in the Drosophila genome that show continued protein coding conservation past the stop codon. And we uncovered a whole new mechanism of post-transcriptional regulation that allows you to just, express all of these proteins by suppressing the stop codon here and just reading straight past it. So we found this whole new class of genes. There's like 300 plus in insects, and there's about 10 of them in the human genome. And we also found experimental evidence of these stop read-through genes. You can also go a step further and basically study not just protein coding constraint and protein level constraint, but you can use this to distinguish additional constraints for non-coding regions on top of the coding regions. So you can measure separately the protein coding constraint and the synonymous constraint. That basically tells you if a protein coding region is expected to be at roughly 2/3 conservation, but you see 100% conservation, there's something weird going on there. And what we found is that that something weird is usually indicative of additional elements that are being encoded on top of the protein coding sequence. Okay? So basically the same nucleotide sequence codes for regulatory regions and for amino acid translation. And if you look at the Hox cluster, the homeobox cluster genes that govern the anterior-posterior patterning segmentation of both human and fly and every vertebrate genome, you see that nearly every one of those genes has a lot of additional constraint and a lot of additional regulatory elements. So you can basically estimate the synonymous and the non-synonymous rates separately and basically find regions that are normal, that basically show substitutions, and other regions that are just perfectly fixed. And that doesn't make any sense if it's just protein coding. And indeed, these have all kinds of functions. Everybody with me here? Question?
Like, how does it blow past the stop codon? This is like a splicing.
So you can basically have. So first of all, let me ask the harder question, which is how does translation actually stop at a stop codon? Right? So how does the protein know to stop translating? What you have is mind-boggling. It's a protein that mimics a stop codon that basically can, that mimics an amino acid that can basically, pretend to be an amino acid. That is this translational release factor that basically normally recognizes. So basically that gets in, and instead of having this, stop codon, you basically sort of, release it. So you could have that by partly binding with a different amino acid. So you could basically have a tRNA that has a mutation, for example, in its acceptor site, and that recognizes one of the stop codons. You could have this mimic protein that mimics the structure of a, nucleotide of a stop codon and sort of, gets in and sort of helps that translation. You could have a mutation in the RNA itself that temporarily disrupts that stop codon. And, so there's many different mechanisms that you can imagine. This is still under investigation. Okay. Who feels that they've learned something? Yeah? Awesome. So that's for protein coding selection. Let's now talk about RNA selection, microRNAs, regulatory motifs, and so on and so forth. Okay? So for RNAs are folded in 3 dimensions, in secondary structure and eventually in 3 dimensions. And you can basically build grammars that generate pairs of matching positions. So in other words, if I'm trying to build a grammar that parses that particular sequence, then I will build a grammar that basically says, oh, okay, start with something, and then, create a fork on the road. That's, I take a sequence and I produce 2 sequences. And now generate pairs of flanking nucleotides that are paired. So A with U, G with C, A with U, G with C, and so on and so forth. And then at the end, generate just nucleotides on only one side of it, G, A. So I can basically I can build a grammar that parses any kind of sequence into a paired or unpaired set of positions. And I can build that for one sequence, or I can build that for a whole phylogeny of sequences, where at every position of my grammar, I generate a whole column of the alignment. Everybody with me here? So that is basically a phylogenetic, stochastic, context-free grammar that allows me to now generate entire sequences of the alignment that are paired or unpaired. Okay? And we can use that to now recognize the evolutionary signatures associated with substitutions that preserve the structure. For example, here, the G is paired with a U, and the U is paired with an A. So if I have a paired substitution, that is actually evidence of secondary structure. So instead of just looking at the primary sequence again, I can look at the secondary structure and basically observe that this is an evolutionary signature for constraint at the structure level. You can do the same thing with motifs. We had talked about that at the regulatory genomics lecture, where we basically can look at the conservation of the position weight matrix. You can look at an evolutionary signature for increased constraint within intergenic regions, or increased intergenic versus coding constraint, or increased upstream versus downstream constraint. And these are genome-wide evolutionary signatures that allow you to distinguish true functional regions from functional motifs from non-functional k-mers of the same type. And you can also apply that to individual instances of a site by basically tolerating mutations and even insertions and deletions as long as they preserve the position weight matrix, okay? And you allow for movement, you allow for insertions, you allow for substitutions, as long as the position weight matrix is conserved. And we talked about how we can translate that from branch length to a confidence that basically says what is the number of instances that I would expect in my signal, that I would expect in my noise, and the ratio between the two tells you the confidence with which a particular position is, conserved functionally. Okay? So for microRNA, similarly, there's 700,000 microRNA-like hairpins, but only 60 to 100 true microRNAs. So you need 99.9% specificity, or a 5,000-fold enrichment. And you can build that by studying the pattern of conservation in these microRNAs. And what you see is that they show a very characteristic pattern of conservation, which is very strong conservation for the two arms, but very little conservation for the hairpin and for the flanking regions. And you can use this conservation profile along with additional structural features and other features to basically get this 5,000-fold enrichment for real microRNAs, okay? And you can basically go and discover all kinds of new properties of microRNAs, okay? And I want to now sort of complete this with the frontiers of where we're at in terms of deep learning and machine learning, okay? Namely, as I mentioned earlier, you can study evolution at the level of individual nucleotides, at the level of patterns of constraints, but you can go a step further and now say, instead of measuring function at the nucleotide level, let's measure function at the translation of that nucleotide. And that translation can be either based on what a convolutional neural network might predict as the function of that region based on a DNA language model, or what the predicted expression of that entire region is for the nearby genes, or the protein language model interpretation of that protein sequence, and so on and so forth. Is everybody with me here? So we have now hundreds of primate genomes and hundreds of mammalian genomes, and we can now start measuring evolutionary constraint across all of these species. So you can basically build these massive multiple alignments that have hundreds of species, and you can use 444 mammals to basically have enough power to detect individual positions. And here's one example of a DNA hypersensitive site, which is active in human and it's active in chimp, but completely absent in mouse. And you can basically see here that the nucleotide level constraint in this region shows a blip specifically in the primate alignment of the 200 plus mammals, 200 plus primates, 239 primates, but absolutely no conservation in the 447 mammals outside the primates. And when you look at the alignment of this region, you see exactly the motif for this TAEAD4 regulator that is perfectly conserved. And you even see these changes that are disrupting that sequence in the mouse, which basically explains why that region is not conserved. Everybody can hear? So you can have this extraordinary power to now detect this. And you can compare this with saturation mutagenesis using these massively parallel reporter assays that I'll talk about in a second. Okay? So you can basically do experiments that allow you to test every single substitution, and then you can measure conservation and see how that relates to each other. And what you see is that when the conservation predicts pathogenicity, for example, the Phylogeny score is, when a mutation is known to be pathogenic, then the conservation score is much higher. And when a mutation is known to be benign, then the conservation score is completely symmetric. So you can basically distinguish functional regions from either experiments that are measuring DNA's hypersensitivity, or using these mutagenesis experiments, or using these disease-associated regions. And in every one of these cases, you basically have this extraordinary power of evolutionary conservation to detect constraints. Okay? So as I mentioned earlier, we can basically look at these increasingly higher levels of abstraction, go from primary sequence to evolutionary signatures to regulatory function to predicted gene expression. And you can basically see this in the context of this convergence model, if you wish, that basically tells you how much conservation is there at the level of individual letters. How is that aggregating at the level of an entire enhancer across multiple mutations? How are these enhancers lining up to a specific promoter for the expression of a gene? How are all of the mutations in the protein coding region of a gene accumulating disruptions of its function? How are all of these mutations across individual motifs, enhancers, promoters, exons, adding up to a total burden for a given gene? And how is that adding up across all of the genes in a given pathway? So as we start thinking about these increasingly high levels of abstraction, you can basically start thinking beyond even just the gene. You can think about the entire pathway. You can think about the entire tissue, and so on and so forth, and basically measure constraint at that level. So we saw how we can use deep learning to predict the effect of a particular sequence on regulation. And you can basically say, okay, given the primary sequence that I can feed as an image, if you wish, using 4 nucleotides wide, which tells me which nucleotide it is between ACGT, and say 100 nucleotides long, you can now use that to start predicting whether something is active in, say, neurons or glial cells, or you name it. And you can build such a predictor that tells you, can I predict the epigenomic activity simply based on primary sequence? Why is that exciting? Because I can build that now using thousands of experiments in the human genome, thousands of experiments in the mouse genome, train such a model, and now apply that model to all 400 mammals, right? So now I can start measuring constraint, not at the level of nucleotides, but at the level of predicted function, okay? And here's how the experiments correlate with the predictions. So you can basically take this tool called Informer. You can find the citation here, that basically predicts genome-wide the level of accessibility for DNAs in a particular cell type. In this particular case, CD14 positive monocytes in a female individual. And here, DNAs in keratinocytes, also in a female individual. And you can see that this is extraordinarily accurate. And therefore, we can now start applying it to many, many species. And you can now see, across different regions, brain regions, cerebellum, frontal cortex, etc. What is the prediction for human based on the human primary sequence? What is the prediction for chimp based on the chimp primary sequence? And then you can look for differences. You can see here that the model predicts that the chimp sequence should have an activity, but the human sequence should not. So now you can start measuring conservation at the functional level rather than at the primary sequence level. And indeed, when you do this, you can start measuring tissue-specific differences in the accessibility and start learning what are the biological functions for which we see such differences. What are the regions of the body? And you can see, for example, in muscle here, or in central nervous system or the peripheral nervous system, how are specific regions changing? And we can see here that rapid evolution, as measured by this indirect way of the predicted function of different elements, is in fact very highly indicative of recent adaptations in the human lineage. And you can see that it is associated with increased psychiatric disorder risk based on these rapidly evolving elements that are predicted to overlap regions that are associated with schizophrenia. So you can now start seeing how you can predict human-specific regulatory function for specific elements that are found to be rapidly evolving by measuring how long is that branch length. And those regions are now shown to also be human-specific in their inferred regulatory activity. And you can now see just how powerful all of these alignments are. When you start looking at primate constraint or mammalian constraint, you see that there's a giant enrichment for heritability, for basically how much of the phenotypic variation is captured by the genetic locations overlapping these elements. So if you take now protein coding exons, that's where you would expect most of the heritability to sit. Well, it turns out that these constrained regions are much more enriched in heritability, almost twofold higher than even protein coding exons. So they're tenfold higher than what you would expect by chance. Okay. So basically mammalian constraint is very enriched, but primate constraint is an even more enriched, suggesting again a lot of human-specific constraints in these. So basically primate-specific constraint in these regions that are clearly associated with disease, but likely to be recently evolved even as close as our primate relatives. And you can see here this conserved, not conserved pattern that I showed you earlier. You can basically now see for a given site, for a given exon, you see this very high conservation here for the splice sites, this high conservation surrounding the splice sites, and then you see this, bang, bang, poof pattern of constraint visible by this single nucleotide level constraint. Everybody with me here? Awesome. And you can, of course, see now conservation beyond just primary sequence by predicting not just whether a nucleotide is conserved, not just whether a motif is conserved, but whether the overall function of a whole region is active. And you can use this by taking a small number of species where experiments have been carried out and then imputing the rest of the species using these convolutional neural networks. Okay, so you can predict accessibility across all of these genomes, and you can use that to now start comparing it, for example, with these high throughput experiments. And I mentioned before these massively parallel reporter assays. So let's dive into them for a second. So you can basically measure not just one region at a time, but you can measure hundreds of regions at a time. How? By taking regions from the whole genome, fragmenting them, and then cloning them into these small plasmid constructs that have a promoter, an enhancer, that have some enhancer, some promoter, and then a test sequence that you're then asking whether it's driving transcription or not. And that basically means that you can simply measure a barcode associated with transcription and then know which of these fragments was actually responsible for that. So there's many different ways of using this technology. The first is you could say, let's take the wild type sequence and then let's mutate it in a small number of regions. So you can take, for example, 5,000 regions and then mutate every single one of these positions. So that's, version number one, where you can basically take activator motifs, repressor motifs in two different cell types, and then see what is the impact of making those mutations. You can go a step further. Instead of testing a single position, you can now offset them by 30 base pairs at a time. So you can basically test 135 base pairs, which is basically what you can fit in your array for synthesis. And you can see that the regions that contain the regulatory motif are in fact showing increased activity as soon as I overlap them, and then decreased activity as I go past them. So that allows me to now do fine mapping at the resolution of 30 base pairs. But you can go a step further and basically say, well, what if I made smaller tiles? Then can I detect with very high resolution exactly when my regions start including that motif and when they start excluding it again? And you can do that for activating motifs, also for repressive motifs, where you see here activity, but as soon as the repressive motif is captured, you see less activity. And you can go a step beyond that and say, well, I can synthesize these regions 145 base pairs at a time, or I can actually just simply cut these regions off of the genome with random fragmentation and then test them using self-transcribing reporter assays. And what that allows you to do is now see in very, very high resolution when the sequences that contain that particular sequence are tested. You see that they're active in yellow much, much more than when you don't have that sequence. And indeed, that's exactly where you see evolutionary conservation. And that's also exactly where you see matches of regulatory motifs. So again, with these tools that we have of evolutionary conservation, with these super high throughput reporter assays, you can basically now start pinpointing individual regulatory elements. And now what you can see is that this, primate conservation predicts these elements much, much more precisely than any other metric. So basically here you see the primate evolutionary conservation and the mammalian evolutionary conservation. And what you can see here for this element that showed a very strong substitution effect when mutated directly using these massively parallel reporter assays. You see this extraordinarily strong effect based on the y-axis being much, much lower. And you see the colors of the primate conservation being very, very strong. So it basically says that primate conserved elements are in fact, much more indicative of gene regulation when tested in the human genome. Okay? So you can now use this to now start measuring these human-specific gene regulatory changes, and you can use this to start predicting evolutionary conservation now at the functional level rather than at the primary sequence level. And the last thing that I want to leave you with is this idea of creating now conservation. Can I use these protein language models that we learned about, in around lecture 9? So you could even use this to actually carry out alignment. And this is a paper by Mona Singh, one of the alumni from here, who's a professor over at Princeton. And what she basically said is, can I take the embeddings, the predicted embedding coordinates of every amino acid, and then ask how conserved are the embeddings of amino acids rather than the primary sequence of these amino acids? So again, you can use these deep learning models either at the nucleotide level or at the protein coding level to basically predict this, higher level of abstraction for the function. And using this, she's able to now show that, this completely new way of doing alignment using the embeddings rather than the primary sequence is, extraordinarily more accurate using very, very little optimization. So you can now basically start aligning every position using this, this transformation of the function. Okay, so that's what I want to leave you with, that basically you can use comparative genomics as an extraordinarily powerful way to detect function in the human genome, that you can find extraordinary resolution overlapping conserved elements, overlapping genome-wide association studies, overlapping these systematic perturbations, where every single time you can measure increased conservation, And you can measure that conservation either at the primary genome sequence or at the primary protein sequence, or you can measure it at the level of evolutionary signatures, the patterns of change, where again, here in white are the conserved positions, which we're not even looking at, and the diverged positions diverge in very different ways in protein coding here in green or in non-coding in red. And that's for protein coding regions, the same thing for RNA structures, for microRNAs, for regulatory motifs across the genome or individual motif instances. And you can go a step further and say, well, beyond just the primary sequence or the patterns of change, I can start looking at the predicted regulatory function using these convolutional neural networks, or even using predicted expression, and higher levels of convergence, if you wish. And same for the primary sequence, you can look at the BLOSSOM matrix, or you can use this iterative blasting, which first searches a sequence, then matches the first matches, and then using that, it can construct a profile hidden Markov model, profile HMM, that allows you to now pay more attention to the regions to the positions that are more conserved in the first matches, and therefore use those to find further matches by refining your model in an iterative fashion. And that's using directly evolution to build this higher level of abstraction, or you can use protein language models that allow you to now take every amino acid and then predict what is its embedding in protein space. And we've also done a lot of work in going beyond just these primary protein language models to now start predicting functional embeddings using knowledge graphs that we're showing here to basically now start predicting not just protein language models that are agnostic of function, but you can actually start predicting the function of a protein using gene ontology, using disease associations, using mutations, using genetics, using expression, using English language of a large language model associated with the description of the function of that protein in the literature. And you can use all of that to start modeling the function of an amino acid in the context of that entire protein, and now start measuring conservation at that level. So this was all about the first part of evolution, where we're using evolution to understand genomes at an unprecedented level of detail. And on Tuesday, we're going to look at how we can use genomes to understand the evolutionary process itself, to study how rapid evolution is happening, to study how new functions are created, to study how things actually change across species. Everybody excited? All right, that's where we'll stop. Thank you.